Hello and welcome back to another notes video. This notes video is your first notes video of Unit 6. This unit is all about DNA. So we're going to talk about the structure of DNA in our first notes. Let's go into a little bit of the history. So this guy named Erwin Chargaff, in that very handsome picture off on the right, was responsible for determining base pairs. So what happened was he was looking at the letters in DNA and he noticed that certain letters fit together. For example, adenine and thymine, A and T, and cytosine and guanine, C and G. So he's responsible for discovering complementary base pairs in DNA. This other lady, Rosalind Franklin, really should have gotten the credit for discovering the structure of DNA. Remember, DNA is microscopic. It's found in our nucleus. So once we had a microscope, it doesn't necessarily mean we know what it looks like. We knew it existed, just not what it looks like. So what she did is she did an X-ray of DNA, and that X-ray kind of throws light waves at DNA, and when things bounce off, they're reflected back as white, and when things go through, they're shown as black. So you get a black and white image from this. From this image, she was able to determine that DNA looks like a twisted ladder. Now, because this was happening back in the olden times and women were not as well respected, she wasn't given any credit for this photograph. In fact, the people who won the Nobel Prize for DNA structure was Watson and Crick. I know, another men stealing women's work. So what happened was, Rosalind Franklin's boss actually gave her picture away to these two gentlemen who used it to make a 3D model. Now, the 3D model was the novel idea. However, they got all the information from other people's work. Rosalind Franklin should have been the one to win the Nobel Prize, but instead it was Watson and Crick, those thieves. So now that we know what DNA looks like, right? We took our picture, we made a 3D model, and we figured out that it's a twisted ladder. We're going to talk about those individual pieces that make up DNA. If you think back to Unit 2, all right, Unit 2 is when we first learned about nucleic acids. DNA stands for nucleic acids. Can you remember what the monomer of a DNA is? It sounds very similar to nucleic acid. It's a nucleotide. Each nucleotide is made of a sugar, a phosphate, and a base. Those bases are the letters that we talked about earlier. A, C, G, and T, which stand for adenine, cytosine, guanine, and thymine. Can you remember which ones go together? Which letter does A go with? If you said T, you would be correct. A pairs with T, which means C pairs with G. They always stick together because they fit together the right way. AT, CG. Some teachers like to remember this as all trees can grow. So, those are called base pairs or nitrogen base pairs, complementary base pairs, because they fit together like a puzzle piece. So, let's review our history, shall we? Which one of these people made the first model of DNA, and which one of these people made the first picture of DNA? The picture came before the model. It was Rosalind Franklin who took the first picture of DNA. It was Watson and Crick who made the first 3D model of DNA. Although they're the ones who won the Nobel Prize, which by me, I'm still a little bitter about that. So, what is DNA? DNA stands for deoxyribose nucleic acid. It holds genetic information. We learned about this in Unit 2 when we talked about macromolecules. DNA holds, in those different letters we talked about, is a section of those is called a gene. A gene is what codes for a certain trait. For example, I have the trait of brown hair. That is coded for in my genes. DNA is made of a nucleotide. I think a nucleotide kind of looks like a cheerleader. where there's a pom-pom in the air made of phosphate. The cheerleader herself is sweet as sugar, and her arm out to the side is a base. If you're not familiar with cheerleading, base is one of the positions. So, phosphate, sugar, base is what makes up DNA. These nucleotides stack together on top of one another to form a chain. On the other side, since DNA is a double helix, there are two backbones, there's going to be another chain. The center of those chains are held together with those letters, the bases. So there are two backbones for DNA. The backbones are made of the pom-pom, phosphate, and the cheerleader, sugar. So it's called a phosphate-sugar backbone. Those steps of the ladder, or the rungs of the ladder, are nitrogen bases, A, T, C, G. They fit together like a puzzle piece. If you can think of when you've made a puzzle, those pieces don't always hold together well. If I pick one up, the other piece falls off. They're held together through what's called a hydrogen bond. We learned about this in Unit 2 with macromolecules when we talked about water. Hydrogen bonds are like magnets, so they're stuck together with magnets in the middle, which is a weak bond, but still a bond. DNA is called a double helix because those two backbones, double, are twisted. The backbones are made of sugar and phosphate, and the steps on the inside are made of nitrogen bases. Take a look here. This is showing a zoomed in image on the left of DNA where you have an individual nucleotide. The nucleotides stack on top of each other to make the backbones, and they're connected in the middle by a hydrogen bond. That ladder that you see in the second image is twisted to make our double helix. So, take a look at this nucleotide. Which one of these is the phosphate? Which one is the nitrogen base? And which one is the sugar? I often compare the nucleotide to a cheerleader. I say the cheerleader herself is sweet as sugar. So the blue is our sugar. In the air, the cheerleader is holding a pom-pom. That pom-pom is the phosphate. And out to the side, she has a base. So, take a look at these images again. Where is the nucleotide? If you said the upper left-hand corner, you would be correct. Nucleotide looks like a cheerleader. Pom-pom, phosphate, sugar, and nitrogen base. Which one of these is the hydrogen bond? Remember, a hydrogen bond is kind of like a magnet. It sticks two things together. Because it's a very weak bond, we often draw it as dotted lines. So if you said the bottom left, you'd be correct. Those letters, A, T, C, and G, are held together with hydrogen bonds, like a magnet. DNA is a double helix. Double because there are two backbones, and helix because it's twisted. So the double helix is the very last image. How many backbones did we say DNA had? It has two. There are two strings of nucleotides. So the upper box is pointing to the two backbones. In between those two backbones is our nitrogen bases. Now I said that those nitrogen bases are held together through a hydrogen bond, a very weak bond. The reason we want the bond to be weak is because oftentimes we might need to unzip DNA in order to copy it. So what is DNA replication? Replication means to copy. DNA will need to make copies of itself before it goes through cell division. So whenever your body makes new hair cells or skin cells, your DNA will make a copy of itself. And when I say a copy, I mean an exact copy. DNA replication produces two identical copies. How does it do this? Well, first, DNA unzips itself. Each strand has letters on it, and in between those letters, the hydrogen bonds are broken. We call these two original strands the parent strands. Those parent strands separate and new strands are built using those original letters as the template for new letters. So here we can see our original strand or our parent strand of DNA. It's time to unzip the strand. How we do this is using an enzyme called helicase. Helicase comes in and undoes the helix. It unzips our DNA. Now that the DNA is unzipped, we have two separate original strands. A new enzyme comes in called DNA polymerase. DNA polymerase builds a new strand to match the original one. When I say match, I mean they're matching those letters. Can you remember what goes with A? If you said T, you would be correct. A pairs with T and C pairs with G. They're building a new strand based off of the original one. So AT, CG, TA, TA, and this chain continues until you've built a brand new strand. With the original strand and the new strand, you have a complete set of DNA. But we said that DNA replication results in two copies. You still have, as you can see at the bottom of the screen, the other original strand. So the same thing happens there. DNA polymerase comes in and matches the letters to build a new strand for that one. In the end, you now have two copies of DNA. Each copy has half original and half new. Because it's half original and half new, we call this process semi-conservative. It means that we're keeping half original and half new in each of the new strands. That's all for our notes on DNA today.